What if I told you that in the fight to cure cancer, giving an artificial intelligence more data about a patient actually makes the AI significantly worse at its job?
Yeah, it sounds completely counterintuitive, right? I mean, we live in this era where we are constantly told that data is king.
Exactly. Feed the machine, right?
Right, feed the machine and it gives you the answer. But in the realm of biology, data without discipline is really just noise.
And that noise is exactly what we are cutting through today. Welcome to the Deep Dive. We're exploring the absolute cutting edge of precision medicine today, and we're guided by a really fascinating 2024 paper from Scientific Reports. It's titled Comparative Evaluation of Feature Reduction Methods for Drug Response Prediction.
It's quite a mouthful, but the core idea is brilliant.
It really is. And the mission for you listening right now is to understand how scientists are figuring out the best, I guess you'd call them shortcuts, to make AI smarter, more accurate, and critically more understandable for your actual doctor.
Because, the ultimate goal of precision medicine is zero trial and error. If you are ever facing a serious diagnosis and you need a highly targeted therapy, the standard, try this chemotherapy and let's see if it works approach is just not good enough anymore.
No, absolutely not.
We want doctors to look at your unique molecular profile and know with mathematical certainty the exact right drug that will destroy the tumor on the very first try, while, sparing all your healthy cells.
But getting to that certainty is a massive head-spinning math problem. So, okay, let's unpack this. We have literal mountains of biological data now, and the researchers in this study used a baseline data set. It's called the Cancer Cell Line Encyclopedia, or CCLE.
Yes, the CCLE.
And the amount of data they collect for just one single sample is staggering.
It really is. Just to give you the specific numbers here, the CCLE measures 21,408 different gene expressions for a single sample.
21,000, over 21,000. And, just so we're all on exactly the same page here, when we say gene expression, what exactly are we measuring? Like, are we just reading the DNA?
Not exactly, no. So picture your DNA as the static blueprint locked in a vault.
Okay, got the vault.
Gene expression is measuring what we call the transcriptome. Think of the transcriptome as the active working copies of that blueprint.
Like the RNA molecules.
Exactly, the RNA. They are out on the factory floor telling the cell what proteins to build right now. So measuring the transcriptome tells you what the cell is actively doing in real time.
Okay, so we have over 21,000 active instructions happening in a single patient sample. You would naturally think, great, just give all 21,000 instructions to the machine learning algorithm so it can learn exactly how this tumor works.
You would think that, yeah.
But this is where we hit that roadblock we mentioned. Why can't we just feed the AI everything?
Well, in data science, we call this the curse of dimensionality.
The curse of dimensionality. Sounds ominous.
It is a bit ominous for the math. The problem is the ratio. We have tens of thousands of dimensions or genes, but usually only a few hundred or maybe a few thousand actual patient samples to learn from. And when you hand an advanced machine learning model 21,000 variables, but only give it a few hundred examples, the model basically panics.
Wait, what does the panicked AI actually look like? Does it just crash and give an error code?
No, actually, it does something much worse. It hallucinates patterns.
Oh, wow.
Yeah, it basically memorizes the specific patients in the training data rather than learning the underlying biological rules. We call this overfitting.
So it's like a student who memorizes the exact answers to a practice test, but then completely fails the final exam because they didn't actually learn the subject.
That is the perfect analogy. The model becomes a black box. It makes a guess about a drug, but it can't explain why. And a doctor cannot, ethically or medically, prescribe a toxic chemotherapy drug based on a black box's unexplainable hunch.
Yeah, I wouldn't want to take a drug based on a hunch either. So we absolutely need shortcuts. Let's look at it this way. If you are trying to identify a suspect in a massive, chaotic crowd, feeding an AI 21,000 blurry security camera angles isn't going to help.
Not at all.
Giving it 10 crystal clear verified fingerprints is far more effective. But I have to push back here for a second.
Sure, go ahead.
In biology, doesn't feature reduction, which I know is the technical term for throwing away data, doesn't that mean we're intentionally blinding ourselves? Like, what if gene number 18,000 was the secret key to curing that specific patient?
It's a really natural concern, but feature reduction isn't about blinding ourselves. It's about removing noise and redundancy to find the actual signal.
Okay.
Because biological genes do not act in isolation. They are highly coordinated. Imagine a marching band.
A marching band, okay.
Right. If you know the drum major has started marching forward, You don't need to individually measure the exact foot speed of all 50 trombone players.
Oh, I see, because you know they are moving together.
Exactly. By mathematically combining redundant genes or just filtering out the one standing still, we drastically improve the AI's predictive performance. And, crucially, we preserve its interpretability for the doctor.
I love that marching band imagery. It really makes it stick. So just to do a quick recap and summarize for you listening before we move on.
Good idea.
We've got 21,000 active genes. Feeding them all to an AI causes it to panic and overfit, creating a useless black box. So we have to reduce the features to find the signal in the noise.
Spot on.
So if throwing all the data at the wall doesn't work, how exactly do scientists decide which data to keep? From what I understand, this study basically staged a massive philosophical debate.
They really did. They pitted two main philosophies of feature reduction against each other.
Let's hear the two sides.
On one side, you have the data-driven approach. This is where we let pure math blindly scan the 21,000 genes and find the statistical patterns.
Just trusting the numbers.
Exactly. And on the other side, you have the knowledge-based approach. This is where we use existing human biological intuition, like years of lab research, to guide the AI on where to look.
Okay, so human intuition versus pure math.
Right. And the researchers ran over 6,000 evaluations, testing 9 different methods across these two philosophies.
Okay, so let's explore these 9 contenders in the arena. I know they fall into 2 broad categories. What's the first way we reduce this massive data set?
The first category is called feature selection. And this is exactly what it sounds like. We just pick specific, highly informative genes and throw the rest in the trash.
Brutal, but effective.
Very. They tested 4 methods here. One was using a curated database called OncoKB, which specifically looks at 1,066 clinically actionable cancer genes.
Okay, so that's just looking at the known troublemakers. What else?
They also tested something called landmark genes. This is a highly specific set of 978 genes.
Wait, why those 978? What makes them landmarks?
Well, through extensive mapping, biologists discovered that measuring just these 978 genes allows you to infer the activity of about 80% of the entire transcriptome.
Wow, really?
Yeah, it's like checking the traffic cameras on the 5 major interstate highways.
Yeah.
If they are backed up, you can mathematically infer that the local side streets are probably jammed too.
That is incredibly efficient.
It really is. The third selection method was drug pathway genes, which just looks at the genes known to interact with the specific drug being tested.
Makes logical sense.
And the fourth was highly correlated genes, or HCG. Now that one is purely data-driven. The algorithm just blindly picks the genes whose activity mathematically aligns with the drug's success in the training data without knowing what those genes actually do.
Okay, so that's feature selection, basically just picking the VIPs off the guest list. But the second category of contenders does something totally different. You call it feature transformation. What does it mean to transform the data?
So instead of throwing genes away, feature transformation squishes the 21,000 genes into new artificial dimensions. A classic data-driven method they tested here is PCA, or top principal components.
Okay, heavy math alert for everyone listening. How do we visualize principal components?
Imagine you have a messy 3D cloud of dots floating in the air, shaped like a teapot.
Okay, a 3D cloud of dots shaped like a teapot. Got it.
If you look at it from the wrong angle, it just looks like a random circle of dots. Just noise.
Right.
PCA is a mathematical algorithm that rotates that 3D cloud until it finds the absolute perfect 2D angle where the shadow it casts clearly looks like a teapot.
Oh, I get it. So it captures the maximum amount of variance, the most distinct shape, using fewer dimensions.
Exactly. You're preserving the big picture while dropping a dimension.
That makes total sense. Now, I saw they also tested something called top sparse PCs. How is that different from regular PCA?
Sparsity is a brilliant mathematical trick. Standard PCA blends every single gene together a little bit. But sparse PCA applies a penalty that forces the model to push as many gene values to absolute 0 as possible.
To 0? Why?
It's like a manager telling a room of 100 employees, if your update isn't absolutely critical, stay completely silent. It forces the algorithm to focus only on the loudest, most important signals, making the resulting data much cleaner.
Ah, okay. The silent employees trick. I like that. They also tested autoencoders, right?
Yes. Autoencoders use a nonlinear neural network, literally a mini artificial brain. It forces the 21,000 genes through a tiny informational bottleneck, compressing it, and then tries to reconstruct the data on the other side.
And what does that do?
The bottleneck forces the network to learn the absolute most vital essence of the data.
Okay, so PCA, sparse PCA, and autoencoders, those are the pure math-driven transformations. But what about the biology-driven ones, the human intuition side?
They tested two knowledge-based transformations. One was pathway activities, which groups downstream genes together to calculate a single activity score for 14 major cancer pathways.
Got it. And the other?
The final contender, and pay close attention to this one, is transcription factor activities, or TF activities.
Transcription factors. Let's make sure we define that clearly for the listener. What is a transcription factor?
Well, if the transcriptome is the active blueprints on the factory floor, transcription factors are the floor managers.
The bosses.
Exactly. They are boss proteins that bind directly to your DNA and give the orders to turn specific genes on or off. Instead of measuring the thousands of individual workers, this method calculates an activity score based purely on what the bosses are doing.
That is such a cool concept. So we have our 9 feature reduction methods, our 9 ways of compressing the data. Now we have to feed that clean compressed data into our machine learning model to actually make the drug prediction.
Right, the actual prediction engine.
I know they use 6 canonical models, including Ridge, Lasso, and Random Forest. But I have to ask the obvious question. If we're doing all this prep work, why not just hand the clean data over to the massive deep learning models we hear about in the news, like the ones powering chatbots and generating videos?
I know, it's really tempting.
Yeah.
But it comes down to the brutal reality of medical data. Deep learning requires millions of data points to function properly.
And we don't have that.
No. If you recall, we don't have millions of clinical tumor samples with perfectly tracked drug outcomes. If you unleash deep learning on small medical data sets, you get that severe overfitting we talked about earlier.
The hallucinating student.
Precisely. Plus, deep learning is the ultimate black box. In oncology, an interpretable model like a standard linear regression model is vastly superior because a clinician can look under the hood and trace the exact mathematical logic of why a drug was chosen.
Okay, let's do another quick recap right here to bring everything together for you. We have 21,000 noisy genes. We must reduce them. The researchers set up an arena with 9 compression methods, some relying on human biology, some relying on pure math.
Yep, the setup is complete.
They are feeding this compressed data into interpretable classic machine learning models, the ultimate goal, to see which combination is the absolute champion at predicting if a tumor will die when exposed to a specific drug.
Which brings us to round 1 of the experiment, the lab reality.
Ding, ding, ding. Round 1.
The researchers started testing in a highly controlled environment using something called the PRISM dataset. This consists of 480 cancer cell lines grown in a lab, exposed to over 1,400 different drugs.
And to measure success, they used a metric called AUC. That stands for area under the dose response curve.
Right.
For everyone listening, imagine a graph where the horizontal axis is the dose of the drug and the vertical axis is how many cancer cells survive. If the cancer cells die rapidly at a very low dose, the curve drops steeply, meaning there is very little area under that curve.
That's a great way to visualize it.
So a low AUC means the cancer is highly sensitive to the drug. A high AUC means the cancer is resisting it.
Exactly. And when they ran the 6,000 evaluations on these lab-grown cell lines, a clear winner emerged for the machine learning model. Ridge regression dominated across the board.
Okay, ridge regression. Why? What is its secret weapon?
Ridge regression is a linear model, but it applies a specific mathematical mechanism called L2 regularization.
L2 regularization. You're going to have to break that down for me.
Sure. So remember earlier when we talked about genes acting like a marching band?
Yes, the trombones.
Right. Co-expressed genes move together. Many standard machine learning models hate highly correlated variables. They panic and will arbitrarily delete one, or their math just breaks down.
They don't like redundant information.
Right. But ridge regression's L2 penalty smoothly handles correlated variables by shrinking their coefficients together.
How so?
Imagine 10 people trying to pull a heavy log with ropes. Standard models might put all the tension on one rope, snapping it. Ridge regression forces all 10 ropes to share the tension equally.
Oh, wow.
Yeah, it perfectly matches the biological reality of how gene networks operate together.
That makes perfect sense. So ridge regression is our winning engine. But what was the best fuel? Out of the 9 feature reduction contenders, who won round 1?
In the sterile environment of the lab, the math-driven top sparse PCs yielded significantly better performance than just using all 21,000 genes.
The silent employee's trick wins.
It did. The algorithm that mathematically forced the noisy genes to zero was highly effective. Interestingly, though, the math-driven methods and the biology-driven methods like landmark genes performed quite similarly overall. Pathway activities performed the worst, actually.
Now, here's where it gets really interesting. Okay, so in a petri dish, complex mathematical transformations like sparse PCs are incredibly powerful. But wait, you and I are not simplified petri dishes.
These certainly are not.
A lab-grown cell line is basically a monoculture. It completely lacks a real immune system, right? There are no blood vessels twisting through it, no surrounding fatty tissue. Doesn't this completely change the game?
You've just hit on the greatest hurdle in pharmacological research. Cell lines lack what we call cellular heterogeneity.
Meaning they aren't diverse.
Exactly. A real human tumor is a chaotic ecosystem. It's a messy mix of mutated cancer cells, hijacked immune cells, chaotic blood vessels, structural proteins, and they're all screaming chemical signals at each other simultaneously.
Sounds like a nightmare for an algorithm.
It is. A mathematical pattern that looks perfectly clean in a uniform petri dish often completely shatters when exposed to the sheer noise of a living human body.
Which sets the stage perfectly for round 2, the clinical reality. Because lab results are famous for failing in actual patients, the researchers took these top-performing models and unleashed them on the ultimate boss, real human tumor data.
Yeah, they used the TCGA dataset. This is real tumor samples from 33 different cancer types extracted from living humans, evaluating 20 specific drugs.
Okay, high stakes.
The ultimate test was, could the models look at this messy human data and accurately separate the patients who were sensitive to the drug, meaning the tumor shrank, from those who were resistant?
The stakes are incredibly high here, and the results completely flipped, didn't they?
They absolutely did.
Oh.
The pure math-driven methods that won in the Petri dish stumbled in the human body.
Wow.
The shocking, undisputed winner in real human tumors was the knowledge-based method. Transcription factor activities. It completely outperformed everything else.
The floor managers?
Yes. It successfully distinguished sensitive from resistant tumors for 7 out of the 20 drugs evaluated. Landmark genes and OncoKB actually tied for second.
What's fascinating here is the sheer efficiency of it, the size paradox.
The size paradox, exactly. If you just used all the raw data, you're processing 21,408 features. Even the highly efficient landmark genes use 978 features.
And transcription factors?
Transcription factor activities won the entire clinical competition using only 293 features.
That is just, I love this. Let's visualize this for you listening. It's like a massive corporation is in crisis, and you need to predict if they are going to pivot their strategy.
Right.
Instead of polling every single one of the 21,400 entry-level employees who are just reacting to daily gossip, you simply interviewed the 293 department heads, the transcription factors.
That is a phenomenal analogy.
Because those 293 people are the ones actually writing the memos and giving the orders to the rest of the company.
And if we connect this to the bigger picture, it explains exactly why pure math failed and biology won.
Why is that?
Because human tumors are noisy. Individual gene expressions fluctuate wildly based on tiny environmental changes. A slight change in oxygen, a mild immune response. Pure math gets distracted by that noise.
It's chasing the gossip?
Yes, but a transcription factor represents the functional cellular state. It acts as a biological aggregator. By the time a transcription factor activates, the cell has already synthesized all those noisy signals and made a definitive decision.
It's an incredibly robust signal.
Precisely because it cuts through the biological chaos.
So if you want to predict how a living, breathing human will respond to a drug, don't blindly crunch 21,000 numbers. Look at the 293 biological bosses and let the steady tension-sharing math of ridge regression do the calculation.
Exactly. And this leads us to the critical final phase of the deep dive today.
Yep.
Interpretability in action.
Right, because we established earlier that doctors will not trust a black box.
No, they won't.
Right.
So the researchers had to prove that this winning combination, ridge regression plus transcription factors, actually made medical sense.
They had to open the box.
They had to open the box.
So when they looked inside the model, what did they see? Were the 293 department heads actually making the right calls?
They absolutely were. For example, the researchers asked the model to evaluate sensitivity to a chemotherapy drug called gemcitabine, specifically in pancreatic cancer.
Okay, pancreatic cancer.
When they opened the model, they saw it heavily relied on a single transcription factor known as IRF1 to make its prediction.
Okay, IRF1. And what about for drug resistance?
When evaluating resistance to another major chemotherapy drug, cisplatin, the model flagged 2 specific transcription factors, HOXB13 and NFE2L1.
But wait, how do we know the computer wasn't just hallucinating those names based on a statistical fluke? Like, did those specific proteins actually mean anything to the oncologists?
That is the most validating part of this entire study. Those findings perfectly align with decades of independent biological literature.
Really?
Yes. Completely separate lab studies have already proven that high expression of IRF1 makes pancreatic cancer highly sensitive to gemcitabine. And we already know from clinical trials that an overexpression of HOXB13 actively causes cisplatin resistance.
Wow, that is huge. So the machine learning model didn't just find a random mathematical glitch to get a high score. By being forced to focus on the biological bosses, the AI independently rediscovered, published, peer-reviewed human biology just by looking at the raw numbers.
It learned genuine biology.
Yeah.
And that is the absolute gold standard for an interpretable medical model.
I can imagine.
When a doctor sees that the AI's logic perfectly mirrors verified biology, they gain the confidence to actually use that prediction to guide a real patient's treatment. It ceases to be a black box. It becomes a powerful, trusted clinical tool.
So what does this all mean for you and me? We've figured out the best shortcut. We know human intuition combined with ridge regression beats pure math in the clinic. What is the next step for this technology? How soon does this change the way we are treated at the hospital?
Well, this study marks a vital pivot for the entire field of bioinformatics. It proves that we shouldn't just be chasing bigger, more complex, power-hungry, deep learning algorithms.
Right.
We need to build algorithms that deeply integrate human biological knowledge. The future trajectory is all about upgrading the knowledge we feed into these models.
How do we upgrade it? Like, what's the new data?
Two massive frontiers are opening up. First, single-cell profiling technologies.
Single-cell profiling.
Right now, when we sequence a tumor, we are basically tossing the whole chunk of tissue into a blender and measuring the average.
Sounds messy.
It is. But single-cell technology allows us to see the exact transcriptomes of every individual cell type inside the tumor. The cancer cells, the immune cells, the tissue cells. We can track those transcription factors with microscopic laser precision.
That is incredible. And what's the second frontier?
Mapping post-translational modifications.
Okay, big words again.
No, sorry. These are essentially tiny chemical tags like phosphorylation that attach to proteins and change how they behave after they've been built. As experimental technologies improve our mapping of these tags, the biological intuition we feed into our AI will become exponentially more potent. We aren't just relying on algorithms to save us, you know. We are building a perfect translation engine between raw, chaotic biology and precise clinical action.
It is truly a phenomenal step forward. It means that when you or someone you love needs a targeted therapy in the future, the data used to save your life will be focused, interpretable, and incredibly accurate.
It's a very hopeful future.
To tie a bow on our journey today, let's just trace our steps one last time.
We started with the curse of dimensionality. 21,000 noisy genes causing our AI to panic.
Right.
We witnessed a clash between math-driven algorithms and biology-driven intuition. And we saw the ultimate triumph of transcription factor activities, those 293 cellular department heads cutting through the chaos of real-world human tumors, all held together by the steady, tension-sharing math of ridge regression.
It really is a perfect synthesis of human knowledge and machine capability. But I want to leave you, our listener, with a final, unaddressed thought to ponder on your own. We've just learned that transcription factors are the absolute clearest signal of whether a tumor will resist a deadly drug.
The clearest we have, yes.
If they're the true bosses dictating how the cell defends itself, what if the future of cancer treatment isn't just predicting what those bosses will do?
Oh, I see where you're going.
What if the next major frontier in pharmacology is actively developing primer drugs? Like medications designed exclusively to hack, hijack, and reprogram those specific 293 transcription factors before the main chemotherapy is even delivered.
That raises a really important question about moving from mere prediction to active, preemptive manipulation of the cellular state. It's a fascinating frontier.
It really is. Imagine disarming the guards before the cavalry even arrives. Thank you for joining us on this deep dive into the molecular shortcuts of precision medicine. Keep questioning the data, and we'll catch you on the next one.